Good afternoon, Starshine. So today we'll be going over Day 6, Unit 7, Genetics. Um, so let's get started. AIM. How do mutations arise in an organism? Take this time to pause the video and try annotating the AIM yourself. Okay, so I annotated mutations and organism. For mutations, I know that it's a change in DNA, and I also know that in order for it to be passed down, what makes it hereditary is that it must occur inside of the gamete. I also annotated organism. I know that an organism is a living thing, and I know that I'm a living thing, so I annotated myself. Moving on to the do now. Question 1. Here I see to the left we have a diagram of DNA. I know that it's DNA because I can clearly see that in each base code, in each base pair, there's a T which indicates thymine, which is a base found in DNA only. Okay. Now, in order for me to describe the main function of this process, I have to first analyze what is going on in this diagram. Looking at the beginning of the diagram, we can see that we have a double-stranded, double helix strand of DNA, right? We know it's DNA again because we see the thymine there, and it's double-stranded. Now, we can see at this point here that it becomes unzipped. We know that it is unzipped by DNA polymerase, that is the enzyme that is used in DNA replication. And if we also look at those unzipped strands, or we can also call them the template strands, we see that there is a new strand binding to them, okay? So this diagram here indicates DNA replication. Now I want you guys to pause the video and describe what is the main function or purpose of DNA replication. So DNA must occur in order for mitosis to happen. Okay, again, mitosis is cell division, and cell division is responsible for cell growth, cell repair, and cell replacement. As you can see to the right, I drew a little diagram of cell division, and we see here we start with the parent cell, then we end up with a daughter cell, and each daughter cell is special because it has the same exact genetic information as the initial parent cell. Then those two daughter cells go on to have their own daughter cells, and then the process keeps occurring, right? And that's how the same genetic information is spread. And we all know that this is all an example of asexual reproduction. Right? So this is a very important concept and process. But okay, moving on to number 2. Describe what would be different if the process pictured were transcription. So everyone pause the video, take a moment, and answer number 2. In order to answer this question, you have to know that transcription is the process of DNA changing over into mRNA, okay? Now, if we look at the diagram above, that green neon copied strand would be mRNA if this were transcription, okay? And we would know it would be mRNA because each thymine would be replaced with uracil, okay? So instead of having thymine here, this would be uracil. And just as a reminder, DNA and mRNA, the key difference are their bases. DNA contains thymine and mRNA contains uracil. Okay? Number 3, transcribe the following DNA sequence. So we have ATA, CCA, and CTT. Pause the video now and transcribe, which means you're going from DNA to mRNA, right? So that means we're going to be replacing thymine with uracil. Pause the video and do that now. Your transcribed mRNA sequence should read as followed: UAU, GGU, GAA. Number 4, use the genetic code below to translate the RNA strand from number 3. So essentially what they mean from translate is give us the amino acid sequence. So we're going to find the amino acid sequence using the genetic code, the universal code chart, and we are going to find the 3 amino acids that we coded for in this mRNA strand. Take this time to pause the video and get started. For the first codon UAU, you should have found amino acid code TYR. For GGU, you should have found amino acid code GLY. And for the last codon GAA, you should have found amino acid code GLU. Good job. A mutation is a change in the sequence of DNA that is due to an error in replication. Environmental factors such as certain chemicals, radiation, or even intentionally biologically engineered. Okay, so factors such as pollution, smoking, those are all factors that could onset a mutation. Today we're going to review the different types of mutations and how they can affect each cell. So 1, we have a deletion. A deletion is when a base is lost within the DNA sequence. Number 2, we have an insertion. This is where an extra base is inserted or added into the DNA sequence. A deletion and insertion may cause what's called a frame shift, meaning the reading frame changes, changing the entire amino acid sequence after the affected base. A substitution is where a single base is substituted for another. So sort of like when you have a substitute teacher, right? That means that your teacher isn't in school, so you have another teacher in place of that teacher teach the lesson for that day. If a substitution does not change the amino acid sequence, it is called a silent mutation. Okay? A silent mutation is good. Yay! Down below, you're going to classify in each box if either a deletion, insertion, or substitution, and whether or not a mutation is silent. Whether or not the mutation is silent. You're going to state the effects of the change in the sequence, either no change in amino acid sequence or one change or multiple. So before you do that, we have to start with our original DNA sequence. Down below, I am going to write out the mRNA sequence for this original strand here. Oh, let me get my highlighter. Okay. So based on this original strand here, I'm going to record my the codons for the mRNA strand. Pause the video now and you try it on your own first. For the mRNA sequence, your codon should have read as followed: AUG, UGG, AAC, CGC, UGC, and UGA. The translated tRNA sequence, your anticodon, should have read as UAC, ACC, UUG, GCG, ACG, ACU. And if you also notice, you should have seen that your mRNA sequence here is almost an exact replica of your original DNA sequence. There's only one main difference, and again, that difference is T being replaced with U. Okay? In the original DNA sequence, we had thymine. In our tRNA sequence, we have uracil. So for example, if we look at the original DNA sequence here, we see that TAC is similar to the tRNA strand, which is UAC. All right, the only difference here is the T and the U. Okay, moving on. What we want to do next is find the amino acid sequence. So what I'm going to do is I'm going to do the first one with you guys, and then I'm going to ask you to pause the video and complete the rest of them on your own. So for the first amino acid sequence, I'm only going to read the mRNA strand, okay? So this strand here is what I'm reading in order to find my amino acid sequence. Now, my first code is AUG. So in order to find the amino acid sequence, I'm going to go to my chart, and I'm going to look here where it says first base. My first base is an A, so it's going to lead me to this part of the chart. Now, my second base is a U, so it's going to lead me, if I look here, it's going to lead me to this row, or this column. My last codon, or my third codon, is a G, which leads me here. So my initial amino acid code is MET, aka, or also known as, the start codon. This allows the ribosome to start creating the protein. Reading this codon indicates it's time to start. Now take this time, pause the video, and complete the amino acid sequence for yourself. The remaining amino acid sequence code should be read as follows: TRP, ASN, ARG, CYS, and STOP. The stop indicates and lets the ribosome know that it is time to stop. The protein is created. It is completed. That is what the stop codon indicates. Now that we found the amino acid sequence for the original DNA sequence, you guys are going to pause this video now, and you're going to do everything we did for the original DNA sequence for the following 4 mutated DNA sequences, okay? So for mutated DNA sequence number 1, you're going to do the same exact thing we just did. You're going to look at the DNA sequence here, okay? So you're going to look at the DNA sequence. Then you're going to find and determine its mRNA sequence by transcribing it, okay? Then you're going to find its anticodon, which actually I'll put that in red because it'll be similar to the DNA sequence besides, the uracil change. Then you will determine its amino acid sequence, right, based on the mRNA codons. And then you're going to determine if there will be any effects, and what type of mutation has occurred. Okay, so pause the video now and try your best to complete the first mutated sequence. For the mRNA sequence, you should have found codons AUG, UAG, AAC, CGC, UGC, and UGA. For its tRNA sequence, you should have found UAC, AUC, UUG, GCG, ACG, and ACU. For the amino acid sequence, you should have found MET, or START, TYR, ILE, LEU, ALA, THR, THR. In order to determine will there be any likely effects in this mutation, you must compare the mutated amino acid sequence here with the original amino acid sequence here. Now, if we compare the first amino acid code, we have two met, so far so good. When we move on to the second amino acid codon, we see that in the original we had TRP, and in our mutated strand we have TYR. This is a difference, so there will be likely effects. I will answer my question here with yes. Now, what kind of mutation is this? In order to determine what type of mutation this is, I'm going to compare the sequences. Being that we had a difference, we found the difference in the second amino acid sequence, again the TRP and the TYR, I'm going to look at the first two codons of the original and the mutated strand. When I look here, I see in the original strand we have TAC, and in the mutated strand we have TAC. So far so good. No changes there. When I move on to the second codon, I see that in the first strand we have ACC, and in the mutated strand we have ATC. So I think we found the culprit. Just to be safe though, I'm going to continue reviewing each codon so that way I am accurate in my answer. So if we look at the next codon, we have TTG in the original strand, and we also have TTG in the mutated DNA sequence. The next codon, we have GCG. In the mutated strand, we have GCG. So far, they seem to all have the same codons, besides that second codon. But let's continue. The next one, we have ACG in the original. We also have ACG in the mutated. Last but not least, we have ACT in our last strand, and we also have ACT in our mutated. Being that there are no extra bases, I'm going to assume that the mutation is a substitution. If we refer back to the definition, a substitution is when a single base is substituted for another. If a substitution does not change the amino acid, it is called a silent mutation. In this case, however, it was not a silent mutation because the amino acid codes definitely changed. Please pause the video here and complete the remaining 3 mutated DNA sequences. Only press play when you are ready to compare and check your answers. Good luck and try your best. Find these mutated DNA sequences and determine what type of mutations occurred. Okay, now let's go over mutated DNA sequence number 2. If we look at that sequence, the mRNA sequence should have read as AUG, CUG, GAA, CCG, CUG, CUC, and A. Based on the amino acid sequence, you should have had START or MAT, LEU, GLU, PRO, LEU, LEU. Now, will there be likely effects? You should have said yes, because there was an insertion or a frame shift in the second codon. If you look at the mutated sequence here, in the second codon, GAC, when compared to the second codon in the original DNA sequence, you'll see that we originally had ACC, and in this one we have GAC. The guanine was inserted, which caused a frame shift in the entire sequence. So now we have an extra base, which is what makes it a frame shift. Moving on to DNA sequence number 3, you should have found that in the third codon, TTA, that is where the mutation occurred. If you look at the original DNA sequence, you'll see that the third codon reads as TTG. In the mutated sequence, it reads as TTA. Now, the mRNA codes are indeed different. In the mRNA sequence for the original, we have AAC, and in the mRNA code for the mutated, we have AAU. But if you look at our code chart, you'll notice that AAU and AAC actually code for the same exact amino acid. So here's where we see the mutation insurance in play. For that specific mutation, it still works and it'll still code for the same exact protein. So in this case, there will not be any likely effects, and this mutation is silent and it's a substitution. In mutated DNA sequence number 4, you'll notice that the error or the mutation occurred at the second to last codon, ACT. In the original strand, you'll notice that we have ACG, ACT here, but in our mutated DNA sequence, the last two codons read as ACT, ACT. This will ultimately affect the amino acid sequence, because if you look at the end, we have 2 stop signals. This stop signal indicates that there will be a likely effect. In our original amino acid sequence, we had ARG CYS stop, and in this one we have ARG stop stop. This type of mutation is considered a substitution, but in this case it is not silent because the amino acid sequence is altered. Okay, that sums it up for day 6. I hope this lesson was helpful. If you have any questions, please bring them to tomorrow's live stream. Have a good day. Stay safe.